I'm Burns Hargis, president of Oklahoma State University, and I want to welcome you to this first ever Interact webinar. We're glad you could join us. We are pleased that Interact is now the hub for One Health research at OSU. Merging plant, animal, and human science is a key mission for OSU. By uniting academic and industry expertise on one platform, Interact will speed the process of bringing new treatments to patients. I visited the Interact lab recently, and I was amazed by the work done on a pet cancer patient. Today, we are delighted to have with us Dr. Philip Dormitzer from Pfizer. Thank you for being with us. And we want to offer a huge thanks to the entire Pfizer team for its work on the COVID-19 vaccine. You are truly American heroes. Again, welcome to Oklahoma State University. Interact is proud to bring you this informative webinar.
Interact is a university-wide effort housed in the College of Veterinary Medicine, Stillwater campus. Our college has several faculty members who perform basic and applied research. Under the leadership of Dr. Ashish Ranjan, Interact will provide these researchers an opportunity to translate their work into effective therapeutic approaches. Interact also aims to involve a variety of internal and external stakeholders. For example, last year we signed a memorandum of understanding with Albert Einstein College of Medicine to promote One Health research at Oklahoma State University. This and other partnerships across different disciplines are central to Interact's mission and will help generate funds to perform clinical trials. I would like to thank the deans of various Oklahoma State University colleges for encouraging their faculties to participate in this important effort. Finally, many thanks to Dr. Phil Dormitzer. His leadership on COVID vaccine development is commendable, and we are grateful for his willingness to share the research with us. Our college's contributions to mitigate the impact of the COVID pandemic were recognized by many, including the Washington Post, and his seminar is timely to our ongoing efforts. Thank you.
Thank you all for joining today. I would like to sincerely thank main campus administration, Dean Risco, Dr. Malaire, founding members, and the Internal Advisory Committee for their great support in helping establish Interact. As Dr. Risco and President Hargis indicated, Interact aims to promote One Health research by encouraging new research in clinical medicine utilizing devices, therapeutics, and diagnostic platforms. Towards these goals, we have established the research infrastructure in the teaching hospital to support innovative projects. Under the leadership of Dr. Brandow, we have also put together an impressive list of speakers for the Interact Eminent Speakers series. Please attend those seminars and learn more about the cutting-edge research insights. In the formative years, Interact will aim to bridge the gap between various scientific disciplines on campus and encourage new partnerships. We hope that these will ultimately culminate into an extramurally funded center grant. Finally, thank you very much, Dr. Dormitzer, for serving as an eminent speaker. Pfizer's vaccine has been a lifesaver, and we applaud you for your leadership. I sincerely hope we can host you again soon in person at the OSU campus. Over to Dr. Biggs now.
Good morning. We're excited to have you as part of this inaugural event for Interact. Interact, of course, is our Institute for Translational and Emerging Research in Advanced Comparative Therapy. We're really pleased this morning to have Dr. Dormitzer from Pfizer speak to us. It's a great launch of this event. And as Dr. Ranjan mentioned, we hope you'll join us for additional events in this series. We would like to welcome all of you from around the world. We have registrants from various international locations, and we want to make sure that we recognize some of our Oklahoma dignitaries that have joined us. Thank you so much for taking the time. We want to thank our fellow university researchers, faculty, students for joining us. And we also don't want to forget our Cowboy fans right here at Oklahoma State from across the campus for joining us this morning. I'm Rosalind Biggs. I am a faculty member here at the College of Veterinary Medicine and also a member of Oklahoma Cooperative Extension Service. It's my pleasure this morning to introduce Dr. Philip Dormitzer. Hi, Dr. Dormitzer. Again, thanks so much for being here. I'm going to read your bio, and it's quite impressive so that our attendees can get a little bit of a snapshot before we get started on your presentation this morning. So Dr. Dormitzer, of course, is Vice President and Chief Scientific Officer of Viral Vaccines with Pfizer. And as we all know, Pfizer has done a tremendous job of the research and bringing vaccine to us during this pandemic. Dr. Dormitzer leads the viral vaccines research and development programs, including the Pfizer BioNTech RNA-based COVID-19 pandemic vaccine, as well as influenza vaccine collaborations. The COVID-19 vaccine, as we know, has been authorized for emergency use, and mass vaccination campaigns are in progress really around the world. A perfusion F-based RSV vaccine to protect infants through maternal immunization has been advanced from discovery to global phase 3 as well in pregnant women, as well as a cytomegalovirus vaccine candidate is preclinical. Before joining Pfizer, Dr. Dormitzer held positions at Novartis Vaccine, and that included the head of U.S. research. He was a founding member of the Novartis Viral Vaccine Research Center in Cambridge, Massachusetts. And in 2009, his research team supported the development and licensure of 3 H1N1 influenza pandemic vaccines in what remains the most rapid vaccine response in history. In 2013, his team responded to the H7N9 influenza outbreak by supplying U.S. pre-pandemic stockpile with a vaccine generated from a synthesized virus. Before joining industry, Dr. Dormitzer was an assistant professor of pediatrics at Harvard Medical School and led a structural virology laboratory, which, with collaborators, determined the structure of the rotavirus neutralization antigens. He graduated summa cum laude in anthropology from Harvard, conducted paleontological research in Pakistan and Zaire, obtained a PhD in cancer biology and an MD at Stanford. He completed internal medicine training at Massachusetts General Hospital and completed the Harvard Infectious Disease Program Clinical Fellowship. Dr. Dormitzer, we're very pleased to have you today, and really we're ready to get into the presentation and learn more about the effort that has really a monumental effort during this pandemic time by you and your team.
Well, thank you so much, Rosalind, and I really appreciate the invitation, especially to be the inaugural speaker of this series. Yeah, I'm looking forward to telling you a bit about how we developed the vaccine. So I think if we're ready for the slides, we can start showing them. I think they start off probably with a relatively boring disclaimer slide. I'll give you the title, Application of New Vaccine Technologies to the COVID-19 Pandemic. And there is the disclaimer. If anyone cares to read it, I'm sure it can be viewed, but maybe we can move on. Just as the introductory slide, there's an image of the vaccine coming off the manufacturing line at our Kalamazoo, Michigan fill finish facility. So this is where, I don't know if you saw the news images a while back of the trucks pulling out on that first day, and this is the facility from which they left. And so I'll tell a bit of the story about how we got to that point. Next slide, please. This has been a collaboration with BioNTech, our partner in Germany. And a lot is made about how rapid the development was. And that's part of the story we'll tell, but I think the other part of the story I'd really like to tell is how this sort of sprint to develop a vaccine during the pandemic built on research that actually started many years earlier. So it didn't come out of nowhere. And I'd like to tell a little bit about the story of where this came from. And in fact, the collaboration with BioNTech did not start this year or 2020 with the pandemic, but rather started in 2018. And at that time, we decided that we would collaborate to make a RNA-based influenza vaccine. And one of the things that appealed to us so much about RNA as a platform for flu was the ability to swap antigens in and out. Because with flu, you're constantly changing the vaccine. So the ability to change rapidly and readily could make for perhaps better strain matches, which is one of the determinants of how well flu vaccines work. But that ability turned out to be very useful in January of this year. It was at that time that following the outbreak in Wuhan, China, the China CDC posted the sequence of the SARS coronavirus 2, the causative agent of COVID-19, on the web in the GISA data sharing platform. And immediately our colleagues in Germany at BioNTech began synthesizing the DNA templates for the RNAs. And they actually made several at that time. And of those, one ultimately progressed to become the vaccine. Pfizer got more heavily involved in March. It was by that time we had started to see cases in the US that did not have any obvious link to a traveler or to other known cases, which indicated that this was spreading in ways that we were having trouble tracing. And although certainly when I first heard about the outbreak in Wuhan, I had hoped that this would be contained as the related viruses, SARS, the first SARS, SARS-CoV-1 rather, or the MERS, the Middle East Respiratory Syndrome coronavirus, were contained just through conventional public health measures. But this sort of hard to trace spread made it clear that that was not going to be the case, and that a vaccine would be needed, and that Pfizer's capability of large-scale development, large-scale production and distribution was going to be needed for a global immunization campaign. So in March, we added to our existing collaboration on influenza vaccines, a collaboration on a COVID-19 vaccine. Our teams have been working very closely together for years, or at least since 2018, and things progressed very rapidly. Actually, by April, the first trial started in Germany and in the US by May. We progressed through phase 1 and 2 so that by July, just a few months later, we were in a large-scale, pivotal, global efficacy trial. And by December, we had our first approval in the UK, first emergency use authorization. We don't yet have approval. It's an emergency use authorization for the pandemic. We had our first emergency authorization in the UK, followed later that month by emergency use authorization in the United States. And now, of course, we are in the midst of a global mass immunization campaign. Now, next slide, please. One of the things that really appealed to us about collaborating with BioNTech, in addition to their capability for RNA therapeutics and vaccines, was a shared philosophy. And that is, we thought that some critical scientific questions, even basic ones about what type of vaccine you're going to make, are best answered with clinical data. And that philosophy was reflected in the way we went about testing this vaccine. And in fact, we did not enter the clinic with one vaccine candidate, we entered with 4. And they included 3 different types of RNA: unmodified RNA, like ordinary cellular RNA; nucleoside modified RNA that has I'll say a bit more about this, that can help evade innate immunity; and self-amplifying RNA that actually can replicate. In addition, we had 2 alternative antigens. One was the full-length spike, which you see depicted to the left under a spike protein. but also just the receptor binding domain, which can flip up and down on the molecule, and it's in the up position in green and in the down position in blue. And through that testing, we found that at least at this stage of development, the nucleoside modified RNA provided us with the manufacturing reliability that we needed. It gave us strong immune responses. It was well tolerated. And we found that while both the receptor binding domain alone, which is how the virus grabs onto cells, and it's a very dense source of targets of neutralizing antibodies, it actually elicited quite good neutralizing antibodies, as did the full-length spike. But the full-length spike gave us somewhat better T-cell responses, and we thought it might be more robust to the possibility of variants. And I'll come back to the variants that are emerging of this virus later. So we selected a nucleoside modified RNA expressing the full-length spike protein for further development. The next slide, please. Now I'll talk a little bit about some of the basic science that went into that vaccine. The first is this nucleoside modification. And this is work that originated at the University of Pennsylvania, and instrumental in the work was Katie Carico, who's now actually at BioNTech, and also Drew Weissman. And they found that a modest change in uracil to make it pseudomethyl uracil, which you can see is just a sort of inversion of two atoms, still allowed the RNA to be translated by the ribosome to make protein. However, it made the RNA no longer efficiently recognized by innate immune sensors such as toll-like receptors. Now, when RNA is introduced into a cell, not the normal way coming out of the nucleus, but from the outside, it does trigger innate immunity. It's a sort of thing that a virus does, and so this is a part of our antiviral defenses. And the cell responds by shutting down expression and translation. So by evading detection by these receptors, as you can see in the graph on the left, in which RNA is used to express luciferase, the red line shows the expression level of luciferase with nucleoside modified RNA, and the blue line showing substantially lower expression from non-nucleoside modified RNA. And on the right, you see interferon alpha production, an indication of the inflammatory response. And as you look from the unmodified on the left to in the middle, the pseudomethyl, this is just pseudouridine in this, not pseudomethyluridine as we actually use in the vaccine, you see much lower expression of this inflammatory mediator. So not only do you have better expression, it turns out it's better tolerated as well. Now, this was not originally intended as a vaccine technology. It was intended as a technology to increase expression levels, for example, for therapeutic expression of proteins from RNAs in the body. And you can imagine that reducing innate immune responses might decrease immunogenicity, but as it turns out, the dominant effect is the increased expression, and you actually get better adaptive immune responses when you quench the innate immune responses in this way. Next slide, please. There's another whole stream of science and really industrial application that went into this. And that's depicted in very schematic form on this slide. This slide comes from Novartis. And this was a glimpse of what happened during the 2009 pandemic, where influenza cases peaked, fell off, and you can see the vaccine that was produced and delivered. And you might think, looking at this slide, well, look, the vaccine started being delivered and the pandemic went away. But that, I'm afraid, wouldn't actually be the case, because you note there is no scale on that y-axis. And in fact, the number of vaccine doses delivered when that red line starts taking off was much too small to have had a significant impact on the pandemic, which is not to say that the vaccine did not do some good. It did, particularly not in 2009 during the pandemic, but during 2010, when the virus came back as a seasonal vaccine. People at that time had time to get the vaccine. And I'd note that this was not the first pandemic wave in 2009. This was the second pandemic wave, the first occurring around spring break time, and then the second occurring when everyone went back to school in the fall. So not only did we miss the first pandemic wave, we missed the second pandemic wave as well. Now, during that time, we had already tried some real innovation in how we could go faster. And in fact, I remember I was driving with my family to New York when I got the word from my boss, who was in Italy at the time, that the virus was now in San Diego. And the head of BARDA, which is the U.S. Biomedical Advanced Research and Development Authority, had contacted my boss, and we'd had a contract at that point for pandemic response. He said, well, you need to deliver somewhere on the web, but the sequence has been posted. Now, at that time, the state of the art for starting a vaccine production was essentially to wait at the mailbox for someone to send you a virus, someone from the WHO. And in fact, there's this large, elaborate infrastructure, global infrastructure, to do surveillance, to isolate viruses, to make vaccine viruses for conventional flu vaccines. We decided we're going to try something different then. We're going to try to make the virus ourselves. We tried to just synthesize it. That didn't work, but we could make it from cDNA. And actually, we need to make it GMP as well. At that time, actually, the experimentation that had been done in rescuing viruses at the company had been a graduate student making viruses for mouse pathogenesis experiments. But we used a laboratory from our collaborators at the University of Marburg in Germany, where they have a BSL-4 laboratory. And although it didn't need BSL-4 containment, that laboratory had the highly controlled circumstances that were very much like GMP, or good manufacturing practice laboratories. And as it turns out, at about the same time that we received the virus from the World Health Organization, we had not only rescued a virus ourselves, but we had done so under close to GMP circumstances. Now, we didn't end up using that virus. Since we had the virus that had the very standard provenance, that had a much smoother regulatory path. So we went with that, but it taught us that if we could develop and make GMP a process to make our own viruses in the same amount of time it took the global system to send us a virus in the mail, well, if we weren't starting from scratch, if we figured out how to do this, couldn't we be much faster? Next slide, please. And so that led to a BARDA, that same agency, sponsored collaboration with the Craig Venters Group. And we made a synthetic approach to generating flu seed viruses. At the time, you could either synthesize DNA rapidly, or you could synthesize it accurately, but you really couldn't do both. But really, some of the innovation that came, particularly from the Venter Group, was in how you could synthesize rapidly and accurately at the same time with enzymatic error correction. And at the same time, we had a separate stream of RNA vaccine development, not nucleoside modified RNA, but self-amplifying RNA. And these came together in a process that we developed under a BARDA contract and actually implemented, because in 2013, there was a pre-pandemic outbreak. It was an H7N9 outbreak in Shanghai, China. Again, the Chinese very promptly shared the data on this strain globally through GISAID, and we started synthesizing. And in fact, as I recall, it was on Easter Sunday that we got the message. And on Monday, our colleagues at the Venter Group in California started synthesizing the genes. On Tuesday, the genes had been synthesized. Actually says maybe that was Wednesday. I know it was Tuesday. It was shipped overnight to us in Cambridge by FedEx. And by the next day, we were starting to synthesize, use the RNA to rescue infectious virus, and as a template to make RNA. By a week, we actually had a sequence verified expression of the hemagglutinin. The virus we made went on to actually, again, back in Germany, in Marburg, to create the pre-pandemic vaccine that became part of the US pre-pandemic stockpile. That was a pandemic that didn't happen. It was scary, but that one did not actually materialize. But the RNA that we made, though it was an experimental batch, not suitable for use in humans, it actually contributed to that vaccine response, because we needed to have antibodies against H7, the H7 hemagglutinin. And we got it by immunizing mice with that RNA They produce the antibodies that we then use, and you can see in that Western blot on the right-hand side on the bottom to detect that we had the right hemagglutinin being expressed from the vaccine virus that we generated also synthetically. Next slide, please. So that talks a little bit about how the technologies that went into the RNA part of the vaccine, and you can see that on the right. And I won't go into great detail, but it's a pretty standard RNA except for the nucleoside modification. Another part of the technology that is extremely important is what you see on the left, and that's the lipid nanoparticle, because if you just inject the RNA, it doesn't do that much. You need to get it into cells. And this lipid nanoparticle is another very fancy piece of biotechnology. It actually consists of 4 lipids. They have different roles. There's an ionizable aminolipid that changes its charge at different points in the production process and administration process, so that at one point it encapsulates the RNA, it then protects it, and then after uptake into cells, fuses with the cell membrane after endocytosis to actually deliver the RNA intracellularly. A different RNA makes up most of that outer layer with another RNA that is pegylated, so that it's sort of shaggy, and that coating helps preserve the particle when it is in the extracellular environment, though it has to come off. So it's actually designed to come off during for entry, because the PEG, while it preserves the particle extracellularly, inhibits its uptake. And finally, the 4th lipid, cholesterol, has a structural role, a bit of a lubricant, keeping everything with good physical properties. So that's, again, another stream of technology about formulation science on how you actually deliver an RNA molecule effectively. Next slide. There is another stream of technology that went into this as well, and that's structural biology. The notion that you could take viral vaccine antigens and engineer them to actually make them behave better, not for the purpose of the virus and affecting, for example, cell entry, but to function as vaccine antigens, had its origin, a decade and a half ago, and actually was sort of demonstrated to work initially with another virus, respiratory syncytial virus. These images show the real scientific breakthrough of Jason McClellan and his colleagues at the NIH. There had been 50 years of vaccine development against RSV with repeated failures. And a key discovery was made through the breakthrough structure of a stabilized pre-fusion form of the protein, because this vaccine antigen is, in fact, you can almost think of it as a device that serves to fuse the viral envelope with the cell membrane to deliver viral RNA into a cell. And it does so by changing shape. And I won't go into all the details of how it changes shape, but suffice to say that on the far left is the pre-fusion structure, the active form ready to attack a cell. On the right-hand side is the post-fusion structure after it's done its job, and you can see the molecule has turned itself inside out. If you want to inhibit the function of the molecule, thereby inhibiting the virus, for example, by binding an antibody to it, you want to immunize with the thing on the left, because there aren't that many patches on the thing on the right that are shared. But the problem is it wants to flip. So even if you have a fair amount of the stuff on the left in the beginning, by the time it's in a vaccine vial, it's all turned into the stuff on the right, which doesn't work so well. And what Jason and his colleagues did was not only determine the structure, but show that you could then use that structure to engineer stabilizing mutations to stabilize the structure on the left. And at Pfizer, we took that and we took it a step further on the next slide. And I believe the next slide has some animation. Oh, it's good. Okay, you can see it all there. That's wonderful. And in fact, it was a huge effort to come up with an optimal structure. We made more than 360 versions of that protein, screened them for physical stability in the pre-fusion form, coming up with our final candidate. And when tested in monkeys, the stabilized pre-fusion form in the graph on the right in blue elicited far higher neutralizing titers than the non-stabilized form, which flipped to the post-fusion form, as you can see in green. And that vaccine is now in phase 3 trials, really in sites around the world, being tested in pregnant women as a means to actually protect their infants by transferring antibodies transplacentally. But that principle was applied also, actually not initially for SARS-CoV-2, but for one of its predecessors. On the next slide, we had 2 warning shots before this pandemic. The first was SARS-CoV-1, the SARS pandemic. The SARS epidemic that never became a pandemic because it was contained by conventional public health measures. And then MERS. And it was with MERS, the Middle East Respiratory Syndrome virus, that Jason, same guy, Jason and his team again, applied their structural engineering. You can see in that panel of electron micrographs down at the bottom. On the left-hand side, you see at the top that long, skinny rod, and then the rest of it, there's more globular, kind of triangular molecules. Again, it's a pre-fusion versus post-fusion transformation of a spike protein that affects entry of a virus. The pre-fusion form is that more globular form. That long, skinny spike is part of the post-fusion form. The head kind of comes off of this molecule as it transforms. And again, the idea was to stabilize the pre-fusion form. That was done by what you can see in the upper right. That helix, turn helix, has to straighten out to go from pre-fusion to post-fusion. And by introducing prolines, it was constrained so it could no longer do so, stabilizing the pre-fusion form. And in the graph on the lower right, you can see the immune response elicited, or the antibody response elicited in red by the pre-fusion stabilized form, and in blue by the non-pre-fusion stabilized form. And again, you see that increase in immunogenicity. So these are these multiple strains of technology that went in, synthetic technology, lipid formulation technology, protein engineering, and now we come to how the clinical technologies that contribute to the vaccine development. On the next slide. We had 2 phase 1 studies, one in Germany, led by our partners at BioNTech, and the study in the US that would ultimately grow into the phase 3 study. We applied principles that we'd used in our RSV vaccine development with very rapid, large, first-in-human studies with multiple arms to test many hypotheses simultaneously, testing in younger and older adults, testing, as mentioned before, multiple vaccine candidates, multiple doses, in an adaptive trial design. In fact, if we had more than 70, something like, it was in the mid-70s, the number of arms that study would have had we just tried to do everything without an adaptive component. But we used the initial results that were facilitated by very rapid transportation of samples. We actually had a plane that was devoted to getting the samples back and forth as fast as possible, very rapid analysis, so that we could take the initial results and adapt the study as we went. And this required tremendous cooperation from the FDA, reviewing data, reviewing modifications to plans very rapidly because of the huge need to move quickly. Next slide, please. Here's some of the data from that initial study. These are neutralizing antibody titers elicited in purple on the left from 18 to 55-year-olds and in blue on the right in those 65 to 85 years of age. On the far right, in gray, are the antibody responses of people who actually had contracted infections with SARS-CoV-2, most of them symptomatic of COVID-19. And what you can see is that, in general, the antibody responses to the vaccine are higher than the antibody responses actually elicited by the virus itself. And a very important point here that I'll come back to. The vaccine was administered on days 1, and then again 3 weeks after that. There's not much neutralizing antibody elicited before the second dose. And so take note of that, because it's key as we think about, for example, coverage of variants. So it takes really 2 doses to get strong neutralizing antibody responses. Next slide, please. You also got very strong T cell responses. In the light blue, you have T cell responses from those who've been infected, and in purple, the T cell responses from those who've been immunized. Again, the responses of those immunized are both T helper cells, CD4 positive cells, and cytotoxic T cells, CD8 positive cells, are as great or in many cases greater than those elicited by infection. Next slide, please. The vaccine turned out to be tolerable with mild to moderate injection site pain, some fevers and chills. Generally, a greater reaction, particularly for systemic reactions to the second dose than the first dose. And fortunately, lesser reactogenicity in older adults who are perhaps, depending on their overall health, less able to tolerate a reactogenic vaccine than younger adults. But the reactogenicity was reasonable and milder in older adults. Next slide gives a few details on, well, actually, before we get to that, this study was a phase 1, 2, 3 study. Next slide. And I'm not aware of any other vaccine trials that have actually been phase 1, 2, 3 studies, but this expanded from that initial part with an adaptive design, many groups, to a very large study with a single final formulation and dose level. That trial enrolled more than 46,000 participants, and it is still ongoing today. We'll follow participants for 2 years, and we are continuing to add arms to that study as new questions arise. And it was conducted at 153 locations around the world. So a truly massive study. We achieved good diversity, both in terms of race and ethnicity, in terms of age, and in terms of underlying medical conditions. Next slide, please. And the results were, I have to say, it was a truly wonderful moment when we saw these results emerge when we unblinded for the first unblinding. In red on the graph are those who received placebo, and plotted is the cumulative incidence of COVID-19. In blue are those who received vaccine. And as you can see, for the first 12 days, the two groups track each other. And from that point on, they diverge dramatically. And I'll come back to the observation that they diverge at 12 days. Because recall, even at 21 days, there's very little neutralizing antibody present. So we're seeing efficacy of the vaccine before there are high titers of neutralizing antibodies. And what you see on the right is the actual vaccine efficacy, and you see it's approximately 95%, and that holds basically true to within a few percentage points across age ranges, across racial and ethnic differences, and is also true with a number of comorbidities. So next slide, please. We got more data on reactogenicity. We see pain at the injection site is reasonably common, but mild to moderate in general. Not a lot of other local reactogenicity. Next slide, please. Systemic reactogenicity, you have the vaccine on the top, placebo on the bottom, also generally mild to moderate, but people do feel somewhat fatigued and sometimes a bit of a headache, some fever, not a tremendous amount of fever, but some fever, less as you can see in older adults than younger adults, and some chills. But within the tolerable range and within the range of other vaccines in common use. Next slide. One useful thing about that reactogenicity is its time course and that it's not that long. You can see that for both younger and older adults, it peaks on the day after immunization. It's diminished substantially by 2 days after immunization and really is quite modest thereafter. Next slide. So this leads us to the vaccine. It is a 2-dose vaccine, 30 micrograms per dose, administered 3 weeks apart. It is indicated for the prevention of COVID-19 in individuals 16 years of age and older, It was rolled out as a 5-dose vial. It turns out with a low dead volume syringe, you can get 6 doses out of the vial. And so that is how it's used now wherever people have access to the low dead volume syringes. It is distributed frozen. It is distributed at dry ice temperatures, and then can be stored at the point of use for 5 days at ordinary refrigerator temperatures. Just recently, we provided a data package to the FDA showing that, in fact, the vaccine also can be stored for 2 weeks at ordinary freezer temperatures, which gives more flexibility in use, and the FDA has approved the transportation and storage for 2 weeks at -20, in addition to these storage conditions. This could be particularly useful in more rural areas or in parts of the world where maintaining an intense cold chain is difficult, though the logistics group has done a remarkable job of being able to distribute, certainly to distribution centers, on dry ice with a very high success rate of delivering the vaccine in good condition to the sites of further distribution. And next slide, please. Now the vaccine campaign is underway, and this slide is now a bit outdated. It says that 12 million people in the US have received either 1 or 2 doses. I checked yesterday, I think we're up to 14 million in the US, and that is, of course, a fraction of the vaccine that has been produced. Much of it is being released, vaccine campaigns are ongoing in the US and now in many other parts of the world as well. To date, the safety that we have seen with real-world use is roughly, in fact, very close to what we saw in the controlled phase 3 trial. So we have not, with widespread use, seen a major change in the safety profile. And in general, most of the reactions that are seen after the rollout of a vaccine or types of reactions are recognized within 2 to 3 months of vaccine rollout. So we're getting past the point where it is likely that new effects will be discovered, because most of them, just from historically, have turned up in the first 2 to 3 months after rollout. Next slide, please. So I'd like to say a few words about the emerging variants. And obviously, there's been a lot written about these. Among them are the UK variant, the South African variant, and the Brazilian variant. The virus does change over time. It does accumulate mutations, including some at the receptor binding site, which is thought to be an important target for neutralizing antibodies. But data I'll show make me optimistic that while we do need to prepare for the potential emergence of a variant that does escape vaccine-mediated immunity, it's not clear to me that we've seen that variant yet, and I'll show you why on the next slide. These are some real-world effectiveness data from Israel, where they have rolled out the vaccine very effectively with a very high proportion of those in their 60s and above immunized, and a good proportion of those below their 60s as well. And I apologize for the small print. But we can see how is the vaccine doing with real-world use. And in Israel, most of the virus circulating is the UK variant. And you can see along the bottom row, against documented infection, we have a 92% efficacy. Against symptomatic illness, 94%. Against hospitalization, 87%. And against severe disease, and it's a whole small intermediary here, I believe it's 92% again. Numbers very similar to what we're seeing in the controlled phase 3 trial. And by the way, these are data taken not so long after that second dose is rolled out. So it is possible that they could even improve further with further observation as you give more time for the vaccine-mediated immune response to develop. And this tells you that, in fact, the vaccine covers the UK variant very well. On the next slide, we have some in vitro neutralization data versus the the South African variant on the right in red versus neutralization of the baseline. This is the Washington strain, the one one of the first strains introduced to the US, on the left, and there is a decrease in neutralization. However, every serum of BNT162b2 immunized people, each of those sera neutralizes the virus. And it's important to note that the level of neutralization you see against the South African variant on the right is substantially higher than the level of neutralization seen after the first dose of vaccine, where we saw protection in our phase 3 trial. So we think it is likely that the vaccine will protect against even the South African variant, which is the most distant variant that we have thus far, though we need clinical data to be sure. Laboratory measures of immunity can give you clues as to what a clinical efficacy will be, but you do have to actually make clinical observations to be sure. Even though we think it is likely that the vaccine will cover, we need to be prepared because we now know that this virus is changing, and it's changing in ways that do affect immune responses. And so we are in fact making a what we call a prototype vaccine to a variant strain, and advancing that to at least a phase 1 safety and immunogenicity trial. Not because we think we need to change right now, but because we need to demonstrate what a change looks like. We have to go through the manufacturing steps, see clinically how does the new vaccine behave, and also smooth the regulatory path so that if a new variant emerges and we need to move fast, we have everything in place to move very quickly, taking advantage of the flexibility of the RNA platform to make an updated vaccine. Next slide, please. And we also have another card to play, and this comes from findings back from pre-pandemic studies with H5N1 highly pathogenic avian flu. In the graphs on the left, you see the immune response to primary immunization. Turns out that wasn't H5N1, that was H5N3, so a strain from quite some time back. And you see that after the 2 doses indicated by the arrows, you get a modest immune response just touching that dotted line, which indicates the 1 to 40 neutralization threshold that people believe may be protective. But if you come back years later with a booster dose, or in this case, 2 booster doses, you can see in the middle and right-hand graphs, you get rapid and far higher immune responses. And as it turns out, those responses are very broad, covering many strains. So one possibility is that if you come back months later with a booster dose for SARS-CoV-2, you'll also get a very high and broad response. We won't know until we test. So we will be testing with both boosting with the updated variant and also with the original variant. So we start to understand what are the rules of immunity for immunization, including booster doses and variant immunization for SARS-CoV-2 as we have learned them for flu. Next slide, please. So based on this, we think there's tremendous promise for the RNA vaccine platform. And we're eager to see what can be done, certainly going back to flu, to see how it does for flu vaccines. Next slide, please. And so our immediate goals are, of course, to continue ramping up production. We've gone from the original projection of 1.3 billion doses produced this year to 2 billion doses. And they're in time now, but we have a network of manufacturing sites across the US and Europe that are working together to increase production. And we're continuing to get higher yields per batch, be able to make more batches per unit time, and exceeding our original production targets. Developing new formulations, trying to get to the point where we no longer have to freeze the vaccine, perhaps by doing things such as freeze drying in the plant so that the distributed vaccine doesn't need to be frozen. We're exploring, as I mentioned, boosting for durability, potentially for breadth. We're studying additional populations. The vaccine is now being tested in adolescents from 12 up. It's being tested in pregnant women. We'll soon be starting a study in children under the age of 12. And of course, preparing for the possibility that an emerging strain could require a vaccine update. It may or may not. We will have to see. Next slide. I'd like to this really does get us towards a vision of being able to use these technologies to monitor viral outbreaks through widespread sequencing, to exchange data rapidly through the internet rather than shipping viruses through the mail, to synthesize our vaccines, and then to have widely distributed manufacturing so that we have the ability to respond much more rapidly and maybe start to catch pandemics in the first waves rather than in the second or subsequent waves. And then the final slide is just there are many people to thank. Of course, the many clinical trial participants and their families, the investigators and contract research organizations and other partners who have helped us develop the vaccine, the government and regulatory authorities who have been working as hard as we are. Operation Warp Speed has been for us a place to exchange knowledge. And of course, the many colleagues at BioNTech and Pfizer who have been really working around the clock over this past year to deliver the vaccine and who are now, particularly at our manufacturing sites, working to get the vaccine produced and distributed. So thanks very much, and I'd be happy to take some questions.
All right, thank you, Dr. Dormitzer. We do have a handful of questions, and we would encourage our attendees, if you're following us on social media live, you can put those in the comment section, and that will show to us, or you can follow your instructions for submission from your registration as well. So give me just a moment. We'll tee up our questions, make sure we're ready to go here. So Dr. Dormitzer, with these emerging strains, and of course your extensive background in vaccines, could you elaborate a little bit more for us? You gave us certainly some hints, but as a community, what does our future hold with COVID? As with the knowledge we have at this point.
Yeah. So it's easy to predict everything, but the future.
Sure.
But there's some sort of different possibilities you can play out. One is the possibility that immunity turns out to be durable and broad, either to the vaccine or to natural infection, and that this becomes like the seasonal coronaviruses. An annoyance, but not that big a deal. And if a vaccine is needed, it would be a pediatric vaccine. A middle possibility is that this becomes a virus that needs a boost every few years, and the rate of change starts to slow down. And this becomes something more like pneumococcus, where every number of years you need either a booster or an updated vaccine that covers the variants that have emerged. And the third possibility is it turns out to be like flu, where it's constantly changing, and every year you're updating and having to repeat immunize. If I were to guess, I would think it's more likely to be not quite like flu. Coronaviruses are different. They have a much lower rate of change. I'm surprised by how much change we've seen. And I wonder how much of it is really being driven by immunity and how much is being driven by this virus still adapting to growth in humans. So we'll find out. I think the key thing is we're going to be prepared for whatever it is we have to do.
Well, certainly that preparedness is going to be important for us moving forward.
For some reason, I don't know if you can still hear me.
Yes, sir.
You can still hear me. So this is a problem on my end. Well, that would be disappointing if I can no longer hear you. Let me see what's going on here.
Can you hear me there, Dr. Dormitzer?
Now I hear you. Yes, I
Okay,
do.
wonderful,
Great.
wonderful.
Okay, good.
All right, so for our next question, talk to us a little bit, and you certainly covered some of this in the presentation, but is there anything more to add on how has the development process for COVID-19 been different, and how does that change us for both? Obviously, you're coming to us from the College of Veterinary Medicine, so those of us in animal health are just as interested in these technologies as well, and really from a One Health aspect, where does that lead us? But how has this development process been different, and how do you see that being implemented moving forward?
Yeah. So, you know, in veterinary medicine and veterinary vaccines, you've been doing this stuff for a long time. I mean, veterinary vaccines are wonderful because the things that in human vaccines you just talk about are being done in veterinary vaccines. I mean, for example, nucleic acid immunization is nothing new in veterinary vaccines. There's a salmon vaccine that's been out for a long time that's used DNA technology, and I believe that there are swine vaccines that are actually tailored to the individual farm with sort of rapid response to the specific strain in the specific facility. So to some degree, we in human medicine, we're learning from what has been done in veterinary medicine. Certainly the notion that when you have to, you can do things in parallel, use adaptive clinical trial designs, do things at risk, are things that can be done in the future if necessary. Now, I don't have an illusion that all vaccine development will look like this, because this has required a singular focus, not just of the company, but of regulators and others, a kind of intensity of focus that in ordinary times can't be done, because it means you have to not do a lot of other things in order to do this. So an important point is no steps have been skipped. Things have been done in parallel. Things have been done efficiently. Decision-making has been accelerated. There's been a very rapid cycle of turning around data and having it reviewed. So the steps are all being done, but what we've shown is when it's really an emergency, things can be done rapidly in parallel and not waiting for one step to finish before starting the next step. For example, scaling up manufacturing before you even know that the vaccine works, which is of course a huge financial risk to build a whole manufacturing infrastructure before you know the vaccine works. But this was a situation that called for that kind of response.
Absolutely, absolutely. You mentioned, we're here with Interact, and we're particularly interested in, our institute here is for translational and emerging research in advanced comparative therapy, hence our acronym Interact. But are there mechanisms where human and animal health collaborations can occur more readily, in your opinion?
Well, yes. I mean, I certainly think of pandemic preparedness, and certainly for flu, understanding the both wild and domesticated poultry disease transmission, as well as in swine, is tremendously important. And the epidemiology has now received the same level of attention that epidemiology of the viruses in humans has. But it's clear that not only is, when it comes to virology, is sort of the global community what we need to worry about, because what is circulating elsewhere comes to us, is absolutely true that the animal community, particularly the animals with which we interact most, we share our viruses with them, so that human health and the animal health are intimately linked.
I think we have time for, I'm looking at the clock here, one more question for you. And I will say on behalf of someone that has family members that are now vaccinated, thanks to the BioNTech team and Pfizer, we cannot say thank you enough and express our appreciation for the efforts that undoubtedly have been round the clock, with probably long evenings and few weekends. So with that, tell us a little bit more about BioNTech. What's distinguishing Pfizer BioNTech from other leading vaccine technologies, and where perhaps do we go from here?
Yeah. So, the partnership with BioNTech has been terrific. I think BioNTech has been an oncology-oriented company, a company that has been very innovative, open to ideas, and has that nimbleness and innovative spirit that comes with biotech. Pfizer, it's a very large pharmaceutical company, has tremendous capabilities, and I've been frankly amazed at how quickly and nimbly it can move, because normally these giant institutions it's kind of like turning an aircraft carrier, but in this case, it has moved quickly, and we've been able to combine the strengths of the sort of very innovative spirit of BioNTech, the capabilities, and under Albert Bourla, who's the CEO, the willingness to take a risk on saying we're going to just do whatever it takes to respond to this pandemic. And so I think the final element of it is that the teamwork was established before the pandemic. And so our research and development teams have been working together for about a year and a half at the time that the pandemic hit. So we already knew how to work together. We knew each other very well. We trusted each other. And that really has enabled us to work together as a very effective team.
Well, Dr. Dormitzer, on behalf of Oklahoma State University and President Hargis, the College of Veterinary Medicine and Dean Risco, and of course, our Interact team here led by Dr. Ron John, we want to thank you again for joining us today. I apologize to our attendees. We're limited with our time we have, but as Dr. Ron John said in his message, hopefully we can have you back here in Stillwater, Oklahoma at Oklahoma State in person in short order, I think is everyone's goal. And because we have definitely some continued interest on behalf of our faculty, as well as those of us that, those that have joined us today as far as attendance. So thank you again. Please express our thanks and appreciation to the BioNTech team and Pfizer as a whole for their monumental effort in this pandemic response. We do want to encourage our attendees to stay tuned for more Interact presentations. And Dr. Dormitzer, I don't think we could have had a better speaker today for our inaugural event. And so thanks again. We do encourage folks to take a look at our social media outlets, as well as follow us online at vetmed.okstate.edu. Under research, you can find our Interact link. You can be monitoring there for future presentations. Our team is already hard at work at getting those lined out for hopefully comparable speakers to Dr. Dormitzer. That will be a hard measure to reach, I am sure. But Dr. Dormitzer, again, thanks so much, and we again want to have you back in Stillwater soon. We'll share some orange power with you on behalf of our Cowboys here. And again, thanks to all those who have taken the time to join us. It will be available at OState TV for those that would like to watch it again. And with that, I think we'll sign off for the day, and hope everyone has a fantastic week and this wonderful start to March. So thanks again, Dr. Dormitzer.